Our first speaker, we're very pleased to have Matt Lauretta join us. Matt is a research biologist in the Sustainable Fisheries Division with the NOAA Southeast Fisheries Science Center in Miami. He holds a PhD in fisheries science from the University of Florida and has studied a diverse range of ecological systems over the last 20 plus years, including pine forests, desert streams, coastal estuaries, and most recently, pelagic fishes. His talk is entitled West Atlantic Bluefin Tuna, Close Kin Mark-Recapture, and this is an area where the Pacific Islands Science Center is keenly interested in potentially delving into, and so we're hoping Matt can help enlighten us. Thank you, Matt.
Well, first of all, I just want to give you guys my sincere condolences for your loss. I know it's hard to carry on. I myself am coming off a pretty strong family tragedy, and I promise you that the hurdles that I present today from a scientific perspective are dwarfed by that loss and coming to terms. So I hope you guys can find the path to the courage to carry on the mission. From what you just told me, it sounds like that's probably what Josh would have liked. So in light of that, thank you all for having me out here. It's certainly a great honor to be here with you in this beautiful place. I'd just like to recognize my international collaborators from around the globe. It's a pretty talented team that I have the distinct honor to sit here and represent their work today. And I especially like to thank our Southeast Science Director, Clay Porch, who was sort of the impetus for all this and had the foresight to get it started and assemble a team to make it happen. So let's start with the basics. You know, capture, recapture has been a goal for fishery scientists for a long time in fisheries assessment to try and estimate the absolute abundance, survival, fishing mortality, growth, migrations. However, it's sort of failed in a lot of its implementation for most programs due to confounding processes of tag loss, handling mortality and behavioral effects from removing the fish from the water, and the imperfect reporting of tags upon recapture, as well as just the fact that doing representative tagging of a population on the ocean scale basin has proved to be infeasible for just about most of the populations, particularly the highly migratory fishes in the pelagic environment. So let's start with what is close kin mark recapture. Essentially, it's a game changer for mark recapture in two ways. One is uses the DNA profiling of individuals to uniquely identify them, a tag that can't be shed. Two, it loosens the assumption that you have to mark and recapture the same individual in the system in order to get the rates of survival and absolute abundance. Now, this is key because you know, when the population has millions of fish, finding that individual back is very challenging. Meanwhile, these individuals, such as siblings and half-siblings, there's thousands of them out there, and they contain essentially the same genomic signal of the spawners that created that cohort. So really, it's a powerful approach to identifying these individuals by the DNA profile of their closely related family members. That's parent-offspring pairs, siblings and half-sibling matches. And essentially, it's a way to mark reproducing individuals in a population based on the genotype in their offspring. The technique has been applied to natural populations to estimate the abundance of reproducing individuals. Successfully applied to southern bluefin tuna has no doubt been a game changer in the assessment and management of that stock. And also evaluated for minke whales. These are highly migratory stocks that have been difficult to assess with traditional methods. But it's not just a technique in wildlife studies. And the following example, I think, is pretty well highlights the power of this tool. That is, when we look at what it's done for crime scene forensics as we moved into DNA profiling to identify individuals and link them to an individual time and place, there is no doubt it's changed the field. And of course, they rely on mark recapture a lot. If you can't capture a culprit at the scene of the crime, then you have to somehow find a way to link them back to it through mark recapture. So there's a lot of parallel between the two fields. For example, if we think about the fingerprint was the primary way to identify an individual for a long time, that's the equivalent of our conventional tag. And if there was a smeared mudge on, then that is the equivalent of a tag loss. Or if the suspect wore gloves, that's like equivalent to a reporting rate issue. Then came video surveillance and automatic face detection, to ability to try and track an individual from the scene to its current location. That's the equivalent of our electronic tagging programs, does the same thing. Then enters DNA profiling, which gives us almost near 100% ability to distinguish an individual in the population. That's the equivalent of sort of the gene tagging approaches that have advanced the salmon world and have been applied to the mackerel in the Southern Ocean. And then we've sort of converged on this one technique now in close kin mark recapture, which now says you don't necessarily have to have that individual back in hand in order to identify who was the individual that you had marked. So there's two general approaches to applying, at least two. I should say that we're going to find more in the future, but for right now, these are the two main ones. One is parentage assignment that's been done for southern bluefin tuna. And with the citation here of Bravington et al.'s work down at CSIRO in Australia. But essentially, here's the general model. You randomly sample the population of adults during the breeding season. This was when they're aggregated by their individual reproductive stock units. Then at some point later, you randomly sample the juveniles after a good period of mixing. You match the parent-offspring pairs to estimate the detection probability of the spawners. And from that, you can divide your catch of the spawning population divided by detection to get you your total population size. And Mark sort of demonstrates this in his diagram here, where all the links between the juveniles are the small fish in the diagram, and the adults are the large one. All the links are the breeding parent-offspring pairs, and the dark ones are your sample. And so you can intuitively think that if you use the juveniles to mark, every juvenile marks 2 spawners. What I love about this approach is it's based on a fundamental law of nature that every single juvenile contains the genotype of 2 parents, 1 male, 1 female, that make up its genome. And so by doing a random sample of each of those populations and looking at the proportion of matches, you actually get the detection probability of the breeding population. The second approach is through SIBSHIP analysis, as was done for minke whales. Here you randomly sample the population of individuals and you estimate the relatedness amongst them as far as half-siblings, siblings, and unrelated pairs. And then you can estimate the breeder population size by those ratios. So again, the foundation behind this is a fundamental law of nature. It essentially says that within small breeding populations, the pool of juveniles will be highly related, and when the breeding population is very large, they'll be highly unrelated, such that you could scale it up. If there was only 2 breeding individuals in the population, then every juvenile would be a full sibling. If there were 4 breeding individuals and breeding was random, then the proportions would be 25% full sibling, 50% half sibling, and 25% unrelated. And it scales up as such as an odds ratio of those. Okay, so application to close kin mark recapture for bluefin tuna. So our objective was simple. It was to estimate the absolute spawning abundance of West Atlantic specific stock by close kin mark recapture. Of course, this has been the simple objective of the stock assessment for over 30 years, and we're still not quite sure that we have it right. But essentially the approach is to take bluefin caught by the US, Canadian, and Japanese fisheries. Those are the 3 major harvesters in the West Atlantic, accounting for over 90%, 95% of the catch. And we're going to genetically compare those to larvae collected from the Gulf of Mexico earlier that year. So we're sort of flipping the equation of Bravington et al.'s approach in the Southern Ocean, and that we're going to use the larvae out in the Gulf of Mexico as a method to mark known active spawners in the system. And I'll demonstrate the study approach a little further in the next slide. But essentially, by estimating the ratio of genetically matched mature fish to young of the year, it's possible to estimate the total abundance of the spawning population for the Gulf of Mexico. Okay, so here's a little more detail. So in order to implement this, we have a long-term monitoring survey for larvae that covers the entire northern Gulf of Mexico during the peak of the breeding season in May. And they encounter anywhere from a couple hundred to 1,000 larvae each year. So under ideal conditions, we could mark hundreds to up to 2,000 spawners every year that are active in the system. We know from pretty intensive satellite tagging out of the Tag-A-Giant and other programs, including NOAA, that fish migrate to the Northeast Atlantic by mid-June, where they mix with the other stocks in the Atlantic Ocean. We know this because this has caused a lot of heartache for the assessment scientists for over 30 years on what we're actually estimating. It's a Western area assessment, and yet we have no idea what the stock of origin for the Gulf of Mexico is. And so then the fisheries operate those three main fleets. They're the hand lines of Canada and the US. They're about 80% of the overall harvest, and the Japan long lines just offshore, and those account for about another 15%. So we're able to meet the mark-recapture model assumptions in that the marked population goes out and mix with the unmarked population. We have really solid evidence from this from about 1,100 satellite tracked fish. 144 of them are known, went into the Gulf of Mexico and resided on the spawning grounds, so we would call them known Gulf origin fish. And none of them died or failed to migrate to this region of the fishery after that. So we have a pretty strong evidence that we're able to meet the mark-recapture model assumptions of the marked population mixing with the unmarked. And then, of course, we do the parentage assignment, and I only use two acronyms in this whole talk. CKMR is close kin mark recapture, and POP is parent offspring pairs, hopefully. And so by doing that ratio, then we can take the total catch. And so we have one key uncertainty is that can we identify Western origin fish in the catches, divide that by the detection probability to determine how many breeders there were. All right, so what I really like about this, and some of you may recognize the form of this equation, is that the model simplicity really is the base. It's really just the Lincoln-Peterson mark-recapture equation with one modification, that we take 2 times the number of juveniles because each one actually tags 2 adults. Take it times the number of adult comparisons and divide it by the observed parent-offspring pairs, and that's our abundance estimate for the number of breeders in the population. And so very intuitively, as the parent-offspring pairs goes up, that implies lower spawner abundance and vice versa. And I'll show you how we can use this framework to do formal hypothesis tests of our stock assessment. Now, the larval marking approach, we struggled with this for some time, whether this would be a suitable marking event, but it has some very solid advantages over, say, like 3 to 4-year-olds, which are being done in the Southern Ocean. One is we have known origin of these fish, right? We capture them within the first week of life on the spawning grounds. So there's no doubt that they're West Atlantic origin fish. We don't have any handling mortality of the spawners because we're actually marking them from their larval genotypes. And this gives us the advantage that nobody handles the fish prior to entering the fishery in the market. So there's no loss of market quality during the marking event. We have no tag loss associated with it because we get consistent genotypes produced in genomes. You can't lose your genetic signal as long as you can get the genetics right. And of course, no natural mortality is our assumption because, as I mentioned, we have a pretty strong indication that all these fish that have none of them, we have no observations of a mortality from a satellite tagged fish leaving the Gulf of Mexico before it enters the fishery. And then, of course, we get the real deal killer has been reporting rates for these large-scale tagging programs because it sort of scales your harvest rate estimate. Could be anything if you don't know reporting rate. Where here is, by doing the larval marking, we actually have no idea what's a marked and unmarked spawning fish until it gets pulled into the dock, genetically sampled, sent to the facility, and the data is returned to us. So there's no way to high-grade tagged or untagged fish from the system. And to boot, we get these extremely useful information byproducts which have sort of caused a lot of uncertainty in the stock assessment, and that is, what is the stock of origin of the US and Canada caught bluefin tuna? You know, obviously our estimates of recruitment in the West Atlantic are biased by migration effects into the system and some unknown proportion of catches over time of East Atlantic fish, of which the stock is 10 times larger, and we have really good evidence that they're migrating over to the west side of the ocean and foraging with these Western origin fish. And then, of course, we get the true fecundity schedule that after a time of doing enough parent-offspring comparisons, we'll know exactly which age classes are producing the juveniles in the Gulf of Mexico. All right, but in order for this to work, we have several core pilot uncertainties that we had to address before we could go into an operational phase. And so these are the study questions the team outlined from the beginning is, do larvae from the Gulf-wide survey provide sufficient quality and quantity DNA for the high throughput genotyping? Can targeted sampling of larval aggregation significantly increase the number of marks, such that when we find an aggregation of larvae, can we go out there and intensively sample it and just get the potential for thousands of marks of the population every year? And then what is the relatedness with both within a toe and between toes on the spatial scale to know really how efficient we are? On the adult sampling side, we need to be able to distinguish east versus west stock of origin in order to move the eastern fish out of the recapture sample. This is the equivalent of having an immigration bias into your marked population. If you don't account for that, you will certainly have a high abundance, a biased high abundance estimate. And then can the existing sampling programs that NOAA has and our international collaborators provide enough numbers of fish to meet the required sample size for a close kin adult estimate? And this is the advantage of this is we already have a lot of money invested out there to sample fish, and if we can just piggyback on that, we save an intense amount of money on implementing field programs, and we can put all the money into genotyping. And then what is the predicted precision based on simulation modeling given our observed sample sizes from the existing programs? So we've now been able to address each one of those questions, and that's why I'm happy to present here today to you that we finally think we can move into the operational phase. On the larval sampling side of things, we found a couple things. One, we lost our historical reference collection. We were hoping to start with larvae collected in 2010 and build that time series of juveniles into the system, because in theory, a lot of those spawners could still be out there and we could push this thing back a decade. But it turns out that they were all stored in formalin and therefore cannot be done for genotyping. So then we had to go push it to the next year. We couldn't use samples we already had. And then we found that the standardized protocol from larval measuring and counts actually rapidly degraded the DNA, that they were having to be sorted in water because they didn't have fume hoods, and that within a few hours under a heat lamp, we lost the genetic signal. So that took us another year to figure out where we were losing the DNA on the larval processing. And then we also discovered that we had this minimum size threshold of about 5 or 6 millimeters, where the proportion of where we could get our DNA concentration above our target threshold. And this is because we were using tails and eyes, because we were piggybacking on existing studies that had the otoliths and gut content allocated. So now we know that we can get them. And then to our second question here, this is the 2016 standardized survey, the location of larvae, where the size of the circle represents the counts of larvae, versus an aggregated effort. When we found them, we asked them to continue sampling that water mass to see if we could increase the numbers. And essentially what we found out is that overall, we were able to get a lot more larvae by targeted sampling. And here you can see we collected 872 versus 342 on the standardized survey. We didn't analyze all of them due to the budgetary allotment, but we found that the amount of fish that we retained, these are the ones that had high enough quality DNA to return the genetic data, that passed the QA/QC from the facility, and then we found a much higher proportional relatedness. So FSP is full sibling pair, HSP is half sibling pair. Here's our total unique spawner genotypes from those two efforts, so about 720 unique fish over 2 years of sampling, and we had a high relatedness, went from about 12% of individuals were either half siblings or siblings up to 50, but what we lost in relatedness, we gained back in DNA quality, such that our mark efficiency was about 40%. What this means that under ideal conditions, if you collected 1,000 larvae, all of them would have high quality DNA, would pass the QA/QC at the genotyping facility, and would be unrelated. That would be 2,000 unique spawner marks. In this case, we have a 40% efficiency, which means for every 1,000 we get, rather than 2,000, we get about 800. But that's still pretty good because they're larvae and they're out there by the tens of thousands. So I'll take 800 marks a year any day for this population. All right, so regarding larval relatedness, during the standardized survey, we actually didn't see any relatedness between samples, and we didn't find any full sibling pairs within samples. It might be a little hard to see here, but essentially there's 2 red points on the left there, up just off the coast of Louisiana, where we did find some half sibling pairs. Compared to when they were following a water mass, they actually found quite a bit of pairs between samples. Those are the dashed lines are half siblings. The solid lines are full siblings, and especially when they got into the aggregation, we found a lot of relatedness. So we know that we're going to lose some on the relatedness, and we have to discount those larvae as not being unique spawner marks. Now, we did have another interesting finding here, is that we actually found some half-siblings across cohorts. What I mean is that samples from 2016 had 2 half-sibling pairs that matched the samples from 2017. This is really interesting for 2 reasons. One, it gives us our first genetic evidence that there's multi-year spawning site fidelity to the Gulf of Mexico. But 2, it also opens up the possibility for spawner survival modeling simply from larval sampling alone, which is a pretty incredible thought, that you could get survival estimates over time without ever handling the adult fish. Regarding the ability to distinguish east versus west stock of origin, this is some work that's been done by our colleagues over at the Spanish Institute of Oceanography that's just been published in Frontiers. We now have at least a 90% assignment to distinguish between the stocks, as well as some unknown assigned fish. And I don't think we're quite sure whether that's potentially a third stock in the ocean or just ones that fell within the 10% error range. But essentially, we see for the first time, this is just a Canadian sample, we have over 4,000 fish in the queue right now at the facility, so we should have some really good estimates all the way down the Atlantic coast. But we're seeing that a high contribution of Mediterranean fish, and it sort of goes up as you move up and offshore, and that's in well agreement with the satellite tagging. Now, what I really like about this, and where I bring my expertise, is in the model simulations. So here, this sort of answers our 2 questions. One, can we get enough samples in from existing sampling programs such that we don't have to implement a large-scale field effort? And I don't know if you can see the sample sizes, but yeah, essentially we were able to get over 1,600 fish in the first year from the existing programs, about, I think it was 1,700, 1,800 the next year, and then we're over at 2,500 in the current. So it's been a slow ramp up and buy-in from the different collaborators, such that now all 3 major nations harvesters are on board to provide their samples from their programs. And then, as I mentioned, I mentioned everything about the larvae to the first 2 years, but incredibly, in 2018, they got about 3,000 larvae in the survey for us. So that's, and if we assume about the same mark efficiency we found for the first 2 years, that's 2,400 adult marks out in the system for 2018. Now, I essentially projected out the stock synthesis model, abundance at age, and kept all the same biological assumptions of maturity, fecundity, and put it into an age-structured simulation. And these are the predicted parent-offspring pairs. What I love about this is it represents a formal hypothesis test of the current stock assessment, such that by the time we get to 2018, if the population is much larger than we think it is in the stock assessment, we would expect the parent-offspring pairs to fall outside this predicted distribution, and that would be strong evidence that we have a low bias in our stock assessment, and vice versa. So this has been really valuable, not only understanding how many samples we need through the simulation modeling, but also to create a framework to formally test our stock assessment. And of course, the leading alternative hypothesis is that it's biased high, so we actually expect much more than that because of the mixing effect of the eastern fish in the western area. All right, just a couple more slides, and then I think we can open it up to questions. But essentially, I started going down this, okay, so now that we're getting close to a population estimate, it's time to start thinking about how to integrate this into the stock assessment. The panel on the right is essentially what the species group thought was the most core uncertainties in the stock assessment. And you can see when we change one little assumption, we get quite a difference absolute biomass scale out of the stock assessment, such that it could be twice the spawning biomass. And this isn't even the full range of them. You can imagine playing with different combinations of indices and such. So I went to it 2 days ago. I said, well, if I'm going to present this, then I probably should test it in the current stock assessment to see how well my first approach here is. So one simple approach I came up with is just put the spawning biomass estimate in there as an index of absolute abundance, fix the catchability coefficient to 1, assign a high weight to it, and integrate it with all the existing indices to see if I could get some scale to the biomass of the spawning population. And sure enough, this isn't a real point, if there's any stakeholders in there, I just picked one that was much lower than the current range of uncertainties to see if I could dial in the stock assessment. And it took some playing with the indices weightings, but essentially by adding an additive process error variance to those other indices of 0.5 CV and a really a low variant CV of 0.1 on this, I was able to get the stock assessment to hit the mark. So that's really promising. The other alternative approach would be just to put a penalty on the likelihood function such that if it wants to push it away from your absolute abundance estimate, which you have high confidence in, it puts a penalty on the likelihood, but that takes recoding of the software, and I wasn't about to start that one yet. So then my vision for this would be actually to use it as a formal harvest control rule on the stock. That is, you have two fundamental pieces of information for managing your resource. What is the trend in domestic stock production? That's absolute abundance estimates from close kin. And then what is the level of resource supplement to the fishery from imported stocks? That's the fraction of the catch that's not of domestic origin. If we put these two pieces of information into a set of logic rules, we can define a priori some rules of harvest control such that when everything's increasing, your domestic production and your supplement, you can increase quota and you can follow the rules of logic, vice versa. And then the argument just becomes by how much, and you can either use a formal change in your abundance in your supplement, or my experience with the management strategy evaluation is that it's actually based on a stability requirement of the fishery to not have wild fluctuations, so that'll be defined between stakeholders and managers. And then I just have a quick list of take-home messages, but I do want to leave some time for questions here, so I won't go through them. But I do understand these slides will be provided to the group, and I just want to say, acknowledge everybody that's really been a huge part of this. Thank you. And then I put some supplemental information for anybody that might be going down this road. These are sort of my lessons learned. There's 2 slides here, so please go ahead and review those and feel free to contact me. But I want to thank you all for your time and attention, and especially having me out here at the center.
So, Matt, if you could kind of encapsulate the question, because we've got a vast audience of people watching online. Kim.
Thank you very much for the presentation. Is this technique limited to species that have discrete mating or spawning areas? And sort of a follow-up, isn't there increasing evidence that there is in fact a third spawning area in the Atlantic?
The question is this limited to species with discrete spawning areas and presumably temporally, and is there definitive evidence for alternative spawning locations in the Atlantic? To your first question, I don't think it is. I think that the goal would be to be able to define those individual breeding units such that you have an idea of how many unique populations there are. I don't think they have to necessarily have a discrete event, but you're probably going to have to do some work on the modeling side of things to account for differential mortalities, presumably, natural mortality. And, but I don't think so at all. I mean, if we look to the literature, you know, tunas are unique, bluefin especially, because they do have a somewhat of a discrete spawning event, but the tropicals, which have protracted spawning, I don't really see that being a deal killer. The key is to get them while they're aggregated in their breeding unit, either the juveniles is the approach we've taken or the spawners themselves, to really identify what's the allele frequency of the individual stock units, and then look for a period of mixing. Both samples don't have to be mixed, but one of them does. Both samples can't be from the aggregated unit because it doesn't meet the assumption that the marked population has gone out and mixed with the unmarked. Regarding evidence for spawning outside the core known spawning regions, we've definitely seen that more recently from opportunistic surveys. They found larvae in the off the continental slope. And what's interesting about that is that little lower pie right there is 7 fish that they caught in their first attempt of the survey, their genetic composition. Some of them were identified with Gulf of Mexico stock of origin, some of them were identified for Mediterranean. Right now, I can't really say that we have a third breeding stock. This gray area certainly could just be the inability to assign an individual because it's not 100% assignment. But I think we're going to get that answer pretty soon. when we get down the road of genotyping our thousands of fish. And it should be any day now returned from the facility.
So you raise an important point that the genetic information that you're collecting for the mark-recapture can also be used for stock identification. So I think that's a real power. But I do wonder, how do you account for the variance in reproductive success of individuals?
That's a great question. It's how do you account for the variance in reproductive success of individuals? Well, that's when you move that Lincoln-Peterson modified equation into an age-structured model, as I've done in my simulations, because you essentially want to account for differential recapture probability on two levels. One is contribution by age class of the spawners, right? So that's just simply a maturity times fecundity type thing times your abundance at age. That all gets put into an integrated assessment as CSIRO does it for southern bluefin. So you actually create a formal age-structured assessment model where you try and estimate those core fecundity maturity schedules to account for differential breeding probabilities. That's inherent in the model simulations that I have, which I'm happy to share with anybody that wants to put their numbers in and their maturity, fecundity, and mortality schedules to get at exactly that. The other level is if you're going to do fisheries recapture sampling, you have to account for unequal probability across age classes by fleet selectivities. That's also put into the model. So those two things interact to create a heterogeneous recapture probability at age, and you want to directly account for that in your model. But some of the interesting results from the simulations is that by the time you combine all these multiple fleets with their different gear types and selectivities, you sort of get a broader schedule. At the end of the day, you can always run a sense of sensitivities on it, differential breeding probabilities or selectivities, and that'll help until you actually can get enough time series to fit the model to those. But certainly that is the way to do it. And but you can start off with your aggregated discrete estimate, and it turns out that from my simulations on Western bluefin, it's not that biased. Maybe 5 to 10% at most, given what we think we know about the reproduction schedules and fleet selectivities.
So thanks, Matt. We're going to try to keep to time, and let's thank Matt again.